It is really my honor to introduce our next award. My former mentor, advocate, and hero, Bridget Hogan, inspired us telling about her journey along a winding road of science. It is particularly exciting that the Society for Developmental Biology recognizes excellence at different points in that scientific journey. We have just heard a wonderful talk from Cagney Kummer about broadening access to that scientific route. The Elizabeth Hay New Investigator Award recognizes a new investigator performing outstanding research in developmental biology during early stages of their independent career. And it is my honor to introduce to you our 2020 awardee, Elizabeth Hay awardee, Celina Juliano, and she fits description perfectly. Celina started her scientific journey as undergraduate student at University of Miami in Florida. She then has done her PhD at Brown University, working with Gary Wessel on germline in sea urchins. It is also notable that as a graduate student, she was involved in deciphering the sea urchin genome. She has done her postdoctoral work at Yale with mentors Robert Steele and Hai-Fan Lin, and this is when her interest turned to Hydra. She started as assistant professor in 2015 in the Department of Molecular and Cellular Biology at University of California, Davis, and her scientific journey really accelerated since she established her own lab. Celine's lab continues to work on PIWI function in Hydra stem cells, and excitingly, her lab has just reported that the PI RNA pathway in Hydra represses transcription element expression in the somatic lineages, and they propose that this contributes to the extreme longevity of Hydra. But in an exciting turn, and in collaboration with a number of labs supported by DARPA, Celina has been involved in leading the effort to decipher the Hydra genome. The end result of this project was the identification of the gene expression states for every cell type in adult Hydra, and also delineation of the developmental trajectories that give rise to these cellular states. And also by performing ATAC-seq analysis, they also identify numerous regulatory elements in this organism. This is a very transformative work that really opened a door to investigating questions that people working in Hydra have been struggling for years. And this is the type of work that the award, Elizabeth Hay Award, is really proud to recognize. Celina, without further ado, we are really excited to hear your talk, and we congratulate you on your achievements, but also we look forward to what you will achieve in the future. Thank you.
Okay, so let me share my screen. All right, wonderful. So thank you for the really kind introduction. And I'm just extremely honored to have received this award from SDB, my favorite society. I've been coming to this meeting for more than 10 years, so this is really fantastic. Really sad not to be with all of you in Chicago, but this online format is really amazing, and I've had a really good time the last few days. So I'd like to say that this award is really a testament to the scientists that I've had in my lab over the last 5 years that I've had the pleasure to work with. And so I really would like to dedicate this talk to them, and I really wish that I was with them in Chicago celebrating. I had dreams of bringing them all to Chicago and having a lot of fun. Hopefully we can do that next year. But for now, I'm just happy to have this opportunity to share with you some of the stuff that we have accomplished and some of our plans. So we are really fascinated by this little freshwater cnidarian hydra, both because it has really powerful adult stem cells, and also because of its remarkable regenerative capacity. And that gives me actually a nice segue just to say it's also an honor to receive an award named after an early pioneer in regeneration, Elizabeth Hay, who did some beautiful work on salamander limb regeneration in the 1950s and 60s. Hydra has been a regeneration model for almost 300 years. When it was first found by Abraham Tremblay to regenerate in 1744. And so I thought it was also a nice occasion to show these original drawings from Tremblay of a regenerating Hydra head right here. So that's the first documented case of Hydra regeneration. And just a quick shout out to Rob Steele, who provided me with that photo. So Hydra is capable of whole body regeneration, meaning that even a small piece of tissue excised from the Hydra, in this case, this is a tissue about 100 microns in diameter, can actually regenerate into a fully formed functional animal. This only takes 2 days in Hydra. So I think that's amazing, and we'd like to know how that works. So Hydra is a cnidarian. So like all cnidarians, it has radial symmetry organized around an oral-aboral axis. This is just a few centimeters high to give you some sense of scale, and it's one of the only cnidarians that's made it into freshwater. So at the oral end, we have the head structures, which include the tentacles and the hypostome. This is where the mouth is. At the aboral end, we have the foot structures, most notably the basal disc, which is a sticky structure that allows a hydra to stick to substrates. The rest of the animal is this body column, which is a hollow tube, and it's the entire body column that serves as the stem cell niche for Hydra. This is where all the stem cells are. And any tissue taken from the body column can fully regenerate, or you can cut the animal anywhere in this tissue and it will regenerate. So Hydra reproduce, when they're happy and well fed, they reproduce asexually, and so we can make clonal populations of Hydra very easily, and this is how we propagate them in the lab. Well, when we coax them, we can also get them to reproduce sexually. I'm just showing here a female and a male that have gone through that are going through gametogenesis, and I would just like you to note that they make few large eggs, right, and they just pop right off the side of their body. They're quite big, 500 microns in diameter, easy to inject. And as it turns out, all you have to do is inject plasmid DNA into the egg, and that integrates into the genome quite readily. And so that was something that attracted me to the model to begin with, this ability to quite easily make transgenics. Okay, so something really cool about Hydra is that it's in this constant state of self-renewal, even as an adult. And in order to really try to explain to you what I mean by this, I just want to talk a little bit about the cell types of Hydra and the cell lineages. So the body of Hydra is really formed by these two epithelial sheets. They're monolayers separated by an extracellular matrix. So this is two different lineages. The outside layer is the ectodermal lineage, and the inside layer in green is the endodermal lineage. So all of the epithelial cells that are in the body column are mitotic, and they're pushed towards the extremities. When they get into the tentacle or hypostome, they'll differentiate into the cells required to build those structures, or if they happen to be pushed down here into the basal disc, they'll differentiate into the cells to build the basal disc. The older cells are then lost, and so in that way, you have this continual renewal. The animal itself is only 3 lineages. The third is the interstitial lineage. These cells are embedded in the interstices of the epithelial cells. So if you take a cross section of the body wall, you'll see all these little interstitial cells in here. In pink, there's a couple different classes of cells that are in the interstitial lineage. These include the neurons, which are in the endodermal layer and in the ectodermal layer, the gland cells, which are in the endodermal layer only, and the nematocytes, which is the stinging cells that typifies the cnidarian phylum, are found in the ectoderm. And all of these cells are supported by a multipotent stem cell, the interstitial stem cell, and this is found in the ectoderm. So the interstitial cells are closely associated with the epithelial cells. That causes a lot of them to get pushed into the wrong place and lost as well, and so they have to be continually replaced. And essentially every 20 days is just an entirely new organism. So this is just to emphasize that we have 3 lineages in Hydra, each supported by a distinct stem cell population. We don't have any totipotent stem cells in the animal. Rather, we have these 3 different types of stem cells, and there's no transdifferentiation between the 3 different lineages. So something that Lila talked about in the introduction is that Hydra has an extreme longevity. I'm not going to talk too much about that today, but I just want to point out that a lot of people call Hydra immortal, which is a cool thing about its biology. Longevity studies have been done by Daniel Martinez, so I put some of these here in case you're interested, and those estimate the lifespan to be hundreds, maybe thousands of years. It's hard to say, that's really difficult to measure, but needless to say, it lives a really long time. And so this ability is likely due to these 3 stem cell populations. And as a postdoc, I was studying the Myb-positive pathway, and this work was continued on in my lab by Brian Tiefy, a graduate student who's getting ready to finish soon. And we found that the Myb proteins are in these somatic cells. For those who aren't familiar with the Myb-positive pathway, it's best known for its function in animal germlines, where the Myb protein with its partner, the small RNA piRNA, can cleave and repress transposon RNAs. So we found that actually the main function of the pathway in the somatic stem cells of Hydra is the same thing, repressing transposons. And we propose this may facilitate the longevity of the Hydra lifespan. Okay. And I'm not going to talk any more about this, but I have these references here if you'd like to take a look. Okay, so something that I really like about Hydra is just that it's quite simple, really. It's not a lot of cell types. It's easy for me to wrap my mind around. So depending on how you were to define a cell type, it has between 20 and 30 morphological cell types. This is a representation here of just all of the different cell types that we have in Hydra. And so it's simple enough that I can just put it all onto one slide. And so when we started up in 2015, we thought, well, it would be useful to just know where all the genes are expressed. And that's a good starting place for any project that we might want to do. In other words, it would be great just to have a complete molecular map of the Hydra. Given its simplicity, it seemed like a possibility. So 3 things came together to make that dream a reality. The first, of course, everybody knows the advent of high throughput single cell RNA sequencing, such as DropSeq, which is what we used. The second was that we had the good fortune of Dr. Stefan Siebert joining the lab for the first 3 years, I guess, 3 or 4 years, 4 years. And he was a project scientist who had the right combination of skills. He had the drive, and he's one of the most careful scientists that I've ever met, and he really tackled this project. And then the third thing that happened is I had the great fortune of becoming friends with Jeff Farrell and talking him into collaborating with me. And this is all through our interactions at SDB meetings. So, that's a plug for going to SDB meetings. Okay, so these 3 things came together, and we single cell sequenced the whole Hydra. We sequenced 25,000 cells from the adult animal using DropSeq. I'm sure everybody's getting really familiar with t-SNE plots, but every dot is cell. They're clustered together based on similarities in their transcriptomes. And we could get markers for all of these populations, do in situ hybridizations, and put labels on all of these cell types. And I want to give a big shout out to Yash Advikun, who was our lab manager for 2 years and who did just a huge number of in situs to help validate all of these clusters. And I'd also like to say that our dream did come true. We now have a molecular map of Hydra. You can go to the Broad single cell portal, and you can go check where all your favorite genes are expressed in our favorite animal. Okay, so of course, what people ask me almost right away is, well, what was the big surprise? Were there new cell types that you didn't know about? What did you discover? I'm actually kind of happy to say that we didn't get a huge surprise. It's not like there was some cell type in Hydra that nobody knew about. And I was happy about this because it meant that these 50 years of work that people have done on Hydra has been largely correct. We know the cell types, we know the lineages, we know how the animal works sort of basically. And now we can get into getting the molecular basis for all of that. But there was one area where we felt, in terms of classifying cell types, we can make an improvement, and that was in the nervous system. So Morphologically, two types of neurons are described in Hydra, the sensory neuron and the ganglion neurons. Of course, we knew molecularly there had to be more based on previous in situ studies. And so we wanted to find out exactly how many there are and kind of make a spatial molecular map of the nervous system. So this project was done by both a graduate student in the lab, Abby Primack, who continues to work on the nervous system, along with Stefan and Yash. Okay, so I alluded at the beginning of the talk that there are neurons embedded in both the ectodermal and endodermal layers, and so we wanted to identify which neurons were in each layer. So we took all of the sequence neurons and progenitors from the data set, and we clustered them here, subclustered them, and you can see that there are the progenitors are in the center. And then we have all the differentiated neurons around the outside. So I don't have time to get into the details, but we did a few other things in sequencing projects in order to assign neurons as being either in the ectoderm or in the endoderm. We ended up finding 9 ectodermal neuronal subtypes and 3 endodermal neuronal subtypes. We then identified markers for all these subtypes and then validated both the fact that they were in the ectoderm or endoderm and also found where they were along the oral-aboral axis using in situ hybridization and transgenic reporter lines. So for the ectoderm, we could do all of this with in situ hybridization. Here's just one example of a gene, HIM176. It's a neuropeptide. Previously had been shown to be expressed in a subset of neurons right above the basal disc in an area called the peduncle. And the HIM176 is in this population of cells, and so we could label these as being peduncle neurons. We did this for all the ectodermal subtypes. But for the endodermal subtypes, we had a difficult time with the in situs, and actually when we went back into the literature, we realized nobody had ever found a marker of endodermal neuronal subtypes. We think it's because it's hard to do in situs on that cell population, and so we ended up making transgenic reporters. So here's one example of this population of neurons called N1. There's a transcription factor, NDF1, expressed selectively here. We extracted the regulatory region, hooked it up to GFP, made a transgenic line, and you can see these beautiful green neurons in the endoderm, but not in the ectoderm. Okay, so now we have this map of the Hydra nervous system, and we have the obvious ability to start driving expression of anything we want in particular subtypes of neurons. So moving forward, we have collaborations with neuroscientists to really establish Hydra as a good model for understanding behavior from molecules to neural circuits to behavior. But as developmental biologists, of course, we're also interested in how the neurons are made and how they regenerate. And so our data set also gives us a really great starting point for all of those kind of experiments. So this is a dot plot put together by Abby, showing every transcription factor in Hydra that has a neuronal expression, and the neuron subtypes are here. And the dots, the size of the dot tells you something about in how many cells we found the transcript, and the color of the dot tells you something about expression levels. So you can see that we have a group of transcription factors that seem to be just required for neuronal identity generally. And then we have transcription factors that are selective for each subtype. So this gives Abby information moving forward for understanding how these neurons are made. And so keep an eye out for work that she will hopefully present at the next SDB. Okay, so of course we didn't just capture cells in the differentiated cells, we also captured cells in the process of differentiating, these different cell states that you can capture, which for developmental biologists, this is a really exciting thing about single cell sequencing. I like to say we try to figure out what's happening with these arrows using our data set. So I'll focus on the interstitial lineage for this talk for the sake of time. So we extracted all of the interstitial cells from the data set and subclustered those. That's what I'm showing here. And we get what we like to call the seahorse structure. And this is really all the differentiation that's happening in this lineage in Hydra. So the stem cells are in the body of the seahorse, and then the cells are differentiating towards into neurons in the head, and they're differentiating into hematocytes in the tail. And there's a lot of differentiation happening. All of these distinct clusters around the seahorse are our differentiated cell populations. I think this really points out how active the stem cells are in Hydra at all times, this huge amount of differentiation that we can see. And so since we have all of these progenitor cells, now we can ask what genes are transiently turned on during differentiation, and particularly we're interested in transcription factors. But in order to more precisely order these cells into a differentiation trajectory, this again is where we worked with Jeff, and he used his software package ERD to build this differentiation trajectory that I'm showing here. So every dot again is a cell, and we are looking at cells in the most naive state down in pseudotime to cells that are fully differentiated. There's again a lot I could talk about and discuss about this differentiation trajectory, but I'll just focus on one aspect for today, which is this was a bit of a surprise to us. We saw this potential state of a neural gland progenitor, so a cell that has started differentiating and has the capability at this point of making a neuron or a gland. It's a putative, this is a hypothesis. We can't say that this is for sure happening, but we do like it in terms of thinking about hydrobiology, because as I mentioned at the beginning of the talk, the interstitial stem cells are in the ectodermal layer, but they must provide stem cells, or they must provide neurons and gland cells to the endodermal layer. And so we know there is some evidence of cell invasion happening here, but we don't know at what state the cell is in when it crosses this ECM. So we are hypothesizing that it's in this, it started to differentiate, it has a potential to make a neural or a gland cell, but it hasn't finished yet, and then it is crossing the ECM. So how do we go into the animal and actually ask if that's true? So the data set allows us to identify genes that are coming on just at the right time. Here's two that we've identified that are transcription factors, MYC3 and then a Myb domain containing transcription factor. So you can see that those are transiently expressed in this potential by potential progenitor, and then it turns off as the cells differentiate. So we did fluorescent in situ hybridization. We could find double positive cells that had the shape of progenitors. And importantly, we could find these cells both on the ectodermal side and on the endodermal side, suggesting that they may actually cross over in this state. So our hypothesis is that these multipotent interstitial stem cells are starting to differentiate. If they stay in the ectoderm, they would just become neurons, but if they cross over, then they could produce neurons, but also gland cells. So we're really interested in this cell invasion event, and in particular, relatively new postdoc Ben Cox is interested in this invasion event, and he would like to understand the genetic and cellular mechanisms that drive progenitor cell invasion. And he will use a lot of different techniques, but something we're quite excited about is that as a graduate student, he had this beautiful paper with live imaging fish scale regeneration. And so he would really like to bring his live imaging skills to this system so we can actually watch this happen. Okay, so I just have a few sort of bigger level slides to go through, and then I'll be done. So we have two sort of big goals in the lab now. One is deciphering the gene regulatory networks that are used to drive homeostatic development, so uncover signaling pathways that turn on transcription factors and lead to cell differentiation. So we're starting with the Wnt signaling pathway because it's the best understood in Hydra right now. We know that it's necessary and sufficient for oral fates, but we don't really know a lot about what it's targeting. And so we have a new student in the lab, Hannah Morris-Little, who has started a project to identify all of the direct targets of Wnt signaling in Hydra. She hasn't been able to go into the lab yet to get to work on it, but she's done a lot of stuff at home already to start answering this question. And then our second big question is to understand then how during regeneration, the conserved injury response is going to activate these pathways to rebuild structures. And this is a question that Jack Cassette, a graduate student in my lab, also one of my first graduate students, is really passionate about and has been working on for the last 5 years. We have just posted a preprint, which I encourage you to check out, And I won't say too much about, there's a lot of stuff in there that I don't have time to talk about, but I will say one thing that we have found in there that's of interest is that these conserved BZIP transcription factors, which are injury response transcription factors, likely activate directly Wnt signaling in the Hydra. Okay, and I also, hey, the good thing about an online meeting is I can advertise a talk that happened 4 days ago. So if you didn't get to see Jack's talk, I highly recommend that you go back and watch the recording. He did a beautiful job, and I'm quite proud of that. And then finally, I just want to touch on this for a minute. A question that I usually get, I'll preempt the question, is, but how good are your genetic tools in Hydra? So the honest answer is they're not great. We can test gene function, but it's slow and it's not efficient, and we really would like it to be better. And so thank you to the NSF for funding a grant in order to help us improve our genomic tools. I'm a co-PI on this with Jacob Robinson. And so we have 2 people in the lab who've been tackling that. Actually, everyone tackles it to some degree, but these 2 are tackling it more. So this is Adrienne Cho. She's our current lab manager. And then one of our new postdocs, Sergio Campos. And I'd also like to say that Sergio recently was awarded a postdoctoral fellowship to study aging in Hydra, which is really what his passion is. So we're excited for him to get that started once we can really get back into the lab. Okay, and the grant also gives us the ability to expand our Hydra community. So please go to openhydra.org to visit our resource hub. We are doing annual workshops so you can learn how to use Hydra. And I'm not going to tell you the dates because everything is up in the air right now, but contact me if you're interested in eventually attending a workshop on Hydra. So I have a few thank you slides. I won't try not to take too long, but this seems like an occasion where I should thank a lot of people. So first of all, I have already thanked and talked about the work of all my lab members. So thank you to them again. I didn't get to mention Robert, who is our first lab manager, who really helped set up the lab. So thank you to him. Didn't get to mention all of our undergraduates, who have also contributed a lot. So Anna, Kevin, Tiffany, Brandon, Elizabeth, and Tika. These are current and former undergrads. Thank you to them as well. Thank you to our funding sources. Thank you, of course, again, to Jeff and Stefan. This is us now. This is us when we used to have fun, and I can't wait until we can do that again. I also just want to give a quick shout out to the Nigerian community. STB is one of my favorite communities, and the Nigerian community is my other favorite community, and they are very supportive, and I love all of them. I want to thank my past mentors. So first, my graduate mentor, Gary Wessel. It's very difficult for me to put into words the things that Gary has given me, but what I will say is that if I could, my goal is to be as good of a mentor to my students as Gary has been and continues to be for me. My postdoc mentor, Hai-Fan Lin, who let me just work on Hydra in his lab, even though nobody else was doing it, and he gave me a lot of latitude, and so I really appreciate him doing that. And then the other thing is my other postdoc mentor, Rob Steele, who taught me, all the stuff I needed to know about Hydra. And he's such a generous person that all it took was me to write one email, and then I had a friend for life. He helped me start working on Hydra. He ushered me into the community, and introduced me to everybody that I needed to know. This is really amazing. And there's just so many people that have helped me along the way. And these are just some of them. Unlike Hydra cells, I can't fit them all into one slide. So these are just some of my scientific colleagues and friends who have helped along the way that I wanted to sort of give a shout out to. And that's where I'm stopping. So if there are time for questions, I'm happy to answer.